Carlos, now you get a little flavor of what we do here on Fridays. Yeah, I'm sure you have something smart to say, but it's more important that we get a chance to just
catch up with one another, for sure.
Honestly, it's the highlight of my week.
Yeah.
So
no,
I'm
it's
glad
really
to invite you to join us and, well, teach us a thing or two.
Perfect. No, I'll just a quick introduction to myself, Joe. So I'm Carlos. I've recently founded a company called MDG CRO, which is a consultancy company and regulatory and clinical research consultancy dedicated to medical devices and in vitro diagnostics. And before that, I still work with a very large medtech consultancy company, RQM Plus, as vice president for diagnostics.
We know them.
And you know them.
I wonder, do you know Allison Komiyama?
Yep, I do. We work closely.
So
Allison was very much focused on the FDA side of things, and I was overseeing everything that has to do with the IVDR and in vitro diagnostics mostly.
Allison won me over immediately when in 2015, she kept her commitment to do a workshop at my conference 3 days after giving birth.
So that's the commitment for sure.
Yes, very much so. And she knew what she was talking about. So yeah, big Allison fan over here.
No, that's good. No, but honestly, Joe, before joining RQM, I was also with the notified body with BSI. I was one of two technical team managers managing a team of technical experts globally and trying to figure out everything that had to do with the IVDR, how we were going to transition from the previous IVD directive to the new regulation, implementing the processes at the notified body level, going through all of the designation in the notified bodies, and really becoming, I believe, was the second IVDR notified body, and we've approved a lot of the first devices that came to market at the time under the new regulation. So I was there with BSI for 8 years. Wonderful experience, but then I decided there was a role for me in consultancy. That's when I moved on from BSI. Before all of that, I'm a scientist, so I used to work within the national health system in the UK in transfusion medicine and transplantation. So usually very close to the operating theater, supporting patients, but also understanding vital parameters and how they progress throughout surgery and that sort of stuff. So
now you told me that it's 5 o'clock by you, so you're no longer in the UK.
I'm no longer in the UK. I'm in Spain at the moment. I live in Barcelona. So I'm originally from Portugal, actually, but I live now between Lisbon and Barcelona. So 2 fantastic cities that I'd recommend. If you haven't been, definitely should be on your list.
I have been to Lisbon and I have been to Barcelona.
Did you like them both, Joe?
Well, I studied a semester in Spain, so I'm quite fond of the area. I remember my trip to Lisbon happened midway through my semester. I was feeling a little homesick and feeling adventurous. We ordered the rabbit off the menu. It was half a rabbit, and it was half a rabbit, like 1 ear, 1 tooth, 1 eye, and I was like, this is not what I expected.
Okay.
And Barcelona was charming, but all I remember is Ramblas. So
okay. No, there's a lot more to Barcelona than Ramblas. That's a touristic area.
I know. Well, I was a tourist. I was a boy. Now I would avoid all of that.
You'll need to come back for sure.
Okay, fair enough. Okay, so teach us something. Oh, by the way, I have to say, you brought your own little posse with you because there are a number of attendees that are brand new names to me I've not seen. And I encourage you all, I've attempted to bring you on screen. I welcome everyone to show their face and ask questions. So I'll invite you again. And if you can, please accept and come on screen. But Carlos, if you have slides or anything, you're welcome to, or if you don't need them, just talk with us and teach us something.
I do have slides. So let me just see if I can work these out because I'm typically on Teams, not on Zoom. So let me just see if I'm sharing the right. What do you see on the screen at the moment?
IVDR clinical performance.
Okay, so that's a good start because it's the right presentation. What about now? Do you see the bit with the notes or without the notes?
I see the webinar one, and I see the next slide with a very handsome picture of you.
So that's not the right one. So let me just change the display settings. What about now?
Now we cannot see the very attractive picture of you.
Okay, that's fine. So that's better actually. You'll see it on the next slide.
Wait a moment.
No, Joe.
I'm going to keep David on mute, so just continue.
That's fine. So the session today really is focused on IVDR clinical performance and It's a presentation I've done a few weeks ago at a conference in Vienna, clinical conference dedicated to both medical devices and IVDs. And it's really focused on some of the strategies to avoid the notified body concerns, because clinical performance, if for the folks on the group that have been paying attention, is one of the biggest gaps at the moment that notified bodies are finding. And I thought it would be nice to put some slides together on how to avoid some of the issues that notified bodies are seeing, both from my experience at the notified body, but also what I've seen through consultancy. And what I wanted to do is like keep the presentation open. So if anyone has questions along the presentation and wants to interrupt me, just feel free, and we'll keep the discussion going. I think that's probably the best approach. Okay. So I've introduced myself, so I'm not going to
very handsome picture.
Thank you.
Needs to be said. Yeah.
And if you allow me just a minute just to talk a little bit about our company, a bit of advertising. I promise it won't take more than 30 seconds. So we are a European-based medtech contract research organization and regulatory consultancy. I'm one of the founders, but I've got a business partner as well, really focused on medical devices. My work is mostly on business development of IVDs, but also providing strategic advice to clients. So we engage in a number of clinical studies for in vitro diagnostics. But also we provide regulatory consultancy, medical writing across a number of areas, from clinical research to regulatory affairs, quality, training, and development of technical documentation. But that's all I'm going to say about my company.
And it's so just at the moment?
There's 8 of us. So we are a small team. So there's me and my business partner, David, and we've got other 6 people in the team.
They're freelancers?
No, they're employed, fully employed by us.
And how old is this group you formed?
So we started this journey 6 months ago, and it's growing. So
yeah, I'd say for you to have 6 full-time salaries and benefits, look at you.
We don't have enough hands at the moment, Joe. So we're looking to recruit more. So we've got a number of projects ongoing, which is good in the current climate. So we feel fortunate that that's the case. But I think it's also a testament to the quality of the work we provide. So
very nice. Please continue.
Okay, so for today's session, so the focus is really on clinical performance under the IVDR. And for us to talk about clinical performance, we need to talk about things such as the intended purpose, what is the depth and extent of performance evaluation, and then the big focus is really on clinical performance, what parameters we should be looking at, some of the lessons learned, looked at near patient tests, use of non-EU data, so those sorts of things. And then I'll finish the presentation with just some take-home messages. But that's just to sort of set the scene of what we're going to be looking at. And one of the things I wanted to start discussing is just a number. So, and I'll start the number with the number 86. And 86 is an important number because it's the number of times the intended purpose is referenced in the IVDR. So just out of curiosity, I think it's one of the top 3 most frequent items referenced in the regulation. So that tells you a little bit why the intended purpose is important. I think it's only overtaken by risk, which is mentioned over 200 times in the IVD regulation. What I would say as well is that when the legislation was first published, there was some confusion around the terms intended purpose and intended use. But notified bodies finally agreed that the terms were meant to mean the same. So if you're looking for IVDR compliance and are worried about the terms intended use or intended purpose, the current interpretation amongst notified bodies is that they are meant to mean the exact same thing. But unfortunately, it's written, some sections of the regulation talk about intended purpose, others talk about intended use, but they're meant to mean the exact same thing. And one of the reasons I wanted to start just discussing intended purpose is because it impacts everything on an application to a notified body. So on clinical evidence, for instance, we've got Article 56 of the regulation that says that the level of clinical evidence shall be appropriate in view of the intended purpose of the device. And with clinical evidence, we've got also, we need to establish a performance evaluation plan that outlines a strategy to prove the intended purpose of a device. So if you define your intended purpose very poorly, it's very difficult to prove the intended purpose and to substantiate your claims in the technical documentation in a way that the notified body will understand it. Also, we've got MDCG guidance on clinical evidence that talks about the importance of the intended purpose, but also GSPRs, for example. Most GSPRs, and in particular GSPR 9.1, which is the focus GSPR on performance characteristics, and 20.4.1 on intended purpose requirements, also provide some of those, some of the importance of the intended purpose, claims, another area, usability of the device. So obviously we need to show that the device works in the context of where it's going to be used. So if it's used by a lay user or in a hospital near where the patient is, usability becomes quite important. So the intended purpose and the definition of that intended purpose is really key for clinical evidence, and in particular clinical performance of a device, because we're trying to prove that the device works clinically for that intended purpose that we've defined up front. And what I wanted to do next is just to give a little bit of the bigger picture under the regulation, and how the different elements of clinical evidence link together. So essentially under an IVDR framework, and if you think of clinical evidence as being the combination of scientific validity, analytical performance, and clinical performance, we need to establish a performance evaluation plan up front that then links to the output of that performance evaluation plan. It's a performance evaluation report, which then links to several other processes from a post-market perspective, such as post-market surveillance reports or PSURs. And then that information is also communicated to users via the summary of safety and performance, the SSP, under the IVDR. So this is just sort of a nutshell of how these processes are interlinked, because it's important to think about the linkages between the different processes, because as you're establishing your performance evaluation plan and your protocols for validating your device, you need to think about what other processes fit within that particular section. So post-market performance follow-up needs to be planned at the stages when you're planning your performance evaluation as well. And then you need to have a PMPF report, a post-market performance evaluation report, around the time when you complete your performance evaluation report. So it's a lot to take into account and to try to map all of these deliverables at the same time. So that just gives you a flavor of the interconnectedness between all of the different processes. So what I wanted to start this presentation with is to look at the depth and extent of performance evaluation. So both through BSI, at BSI and through consultancy, I have seen a wide range of approaches from manufacturers that did too much to manufacturers that did too little on their devices. But the actual IVDR and guidance from the Medical Devices Coordination Group, MDCG, allows for a proportionate approach that can take into account several factors. And two of the most important factors are the novelty and the risk class of the devices. So a notified body will typically expect a lot more for, let's say, a class D HIV assay, for example, compared to a class B type of general physiological marker, like it could be a magnesium test, just to give you an example. But what we also need to take other things into account, such as, are we talking about an assay? Are we talking about a control? Are we talking about a calibrator? So the risk of all of those types of devices also needs to be taken into account. If it's a software, an instrument, who is the intended user of the device? So for a professional use, for example, you know that the people that are going to be doing the test are experienced. So the instructions for use are important, but not as important as they would be for a self-test, for a user that has got no experience in handling IVD. So you'll need to think about providing information that allows for the device to be used safely, versus, for instance, a near patient test. So a test that's used often in emergency conditions. So it could be used in accident and emergency, where there's a lot of stress around the use of that test. So those are some elements manufacturers need to take into account. So the depth and extent really should be proportionate to the characteristics of the device, the risk class, the performance of the device, and its intended purpose. So, and obviously take into account what is currently state of the art for testing a particular condition. So that's just something else that needs to be part of that determination of how much is enough. So in clinical performance, so really clinical performance is defined in the regulation as the ability of an IVD to yield results that correlate with a particular clinical condition or a physiological process or state in accordance with the target population and intended use. And that's why clinical performance, there's a clear link between what you're going to be demonstrating against the intended use that you've defined for your device. So we'll need, the end goal is that we'll need to be demonstrating clinically relevant outputs. So what clinical performance claims are we establishing? So for instance, is the diagnostic sensitivity of 90% appropriate for an HIV test? Probably not, because you don't want to risk a misdiagnosis in 10% of patients that get tested for HIV. So you'll probably be looking at 99% as being state of the art for HIV testing. So that always needs to take into account what is the current state of the art in tests that measure the similar condition, and what the medical decision points are, the cutoff values, things such as the diagnostic sensitivity, diagnostic specificity. And clinical performance really should be demonstrated for the target population that is claimed in the intended purpose of the device, the use conditions, the operating and use environment, and all intended user groups of that device. And that will have a number of implications for the design of the study, of the actual study that you're going to be conducting for demonstrating clinical performance. Don't know if there's any questions up till here. Yeah. If not, so what I want, one of the key questions I get asked all the time is if whether clinical performance is always applicable. So for a lot of manufacturers, it's very difficult to demonstrate clinical performance. And the answer is no, it's not always applicable. And the regulation allows for, in certain circumstances, to say that for my particular device, clinical performance is not applicable. If clinical performance is not applicable, manufacturer won't have to create something required by the regulations, such as the clinical performance report. But remember that even if you can justify that your device, that clinical performance is not applicable to your device, a performance evaluation report is always required. Even if a device does not yield specific clinical claims, the regulation still requires for every IVD out there that a PER, or a performance evaluation report, is always available.
So Carlos, does this differ from the MDR? I work primarily in medical devices and not IVDs, but from my understanding of the clinical evidence and requirements in under the MDR, this is a little bit different. This sounds more like what medical devices used to be able to do under the MDD, where they could write the literature review and essentially like a performance evaluation without actual clinical evidence on their device. So it sounds like IVDs kind of move towards where medical devices came from.
It's a little bit like that, Michelle, but it's also, there's another element to it, is that there are two types of IVDs. There are IVDs that you use to, for instance, do like an HIV test, right? You're actually establishing the HIV status of a patient. And for that, you need to demonstrate that you can establish that IVD status. But there are certain types of IVDs that do not yield clinical claims. So for example, a specimen receptacle, that's, if you think about it as all of you are probably familiar with the swabs that you used to take from your nose when you're taking like a PCR test to do a SARS-CoV-2 assay, right? A COVID test. So that swab itself does not need clinical performance data. And the reason being, because the definition of clinical performance is the association of a clinical condition that the device generates results that associate to a clinical condition. And that is done through the assay that is run after the swab is taken. So the swab itself on its own will not generate results for the patient. So it always needs an assay or a test to be run for the results to be generated. So when you're defining where clinical performance applies is at the assay level. And what you'll say typically is, my swab is a specimen receptacle. On its own, it does not generate results that are clinical, but the clinical performance is established via this assay that is recommended to be used with my swab. So that's more of the rationale. But I think it's just that one of the reasons that's the case is because IVDs are so diverse, and the regulation includes like things like glass slides, microscopic glass slides, or general purpose reagents that on their own, they don't have clinical claims. So often you can say that clinical performance is not fully applicable and can be demonstrated sort of indirectly via the actual device that provides the result to the patient. So, that's more of where the IVDR is coming from. But that's a really good question because we get asked that very, very often, Michelle.
Yeah.
Well,
Yes, thank you, Carlos. I think this is a relevant example. Is this your experience, the swab experience with the notified body already, or is this something like a well understood fact in the industry?
With the notified body already. So there's
Okay.
one thing. So if you think about it, swabs, unless they are sterile, they are not reviewed by the notified body. So only sterile swabs would be reviewed by the notified body.
You answered my second question. So guide wire is the same example.
Yes,
guide wire itself doesn't have, but I think you answered my second question because it's sterile. So they're asking for clinical evidence.
Yes.
Okay.
Carlos, we appreciate that. David,
Yeah, thank you.
So just on top of Goldie there, so they're asking for sterility evidence, not
correct.
The sterility, right?
Yes. So if you've got a swab that is sterile, the only thing that the notified body is going to be looking at is the sterility elements. So they won't necessarily review the clinical evidence around the device. So they're just going to be concerned that you as a manufacturer can ensure sterility of that swab. Because swabs are typically class A devices, so they don't undergo notified body review. If they were sterile, the only aspects that the notified body would be looking at are the aspects related to sterility. So that's the difference here.
So my real question I was going to bring is, what if my swab doesn't pick up anything? What if it's just a piece of silicone or something that is not adherent? It's not fibrous, to grab cells or whatever else. And I get negative test. Don't they care about my collection device, or how does that?
They do. They do. But they will care about your collection device via the test that's used with your collection device. That's how they would get to the manufacturer. Is your test is not providing results. So where is the root cause of that test not providing results? And the manufacturer would need to trace that back to the swab and then implement whatever corrective actions they need to implement. But from a notified body perspective, it would be reviewed on the assay level. So the assay is the bit that provides the result to the patient.
Great, thank you. Joe, I have a lunch meeting I got to leave for. Sorry, this is a great discussion. Thanks, Carlos.
No worries, David.
If you don't have enough time for everybody, David, then it's impolite.
I didn't put out the invite, and it's really business, so
Okay, fair enough. Jan?
Thanks.
My question was the same thing. If it wouldn't pick up the stuff, or the swab head would fall off, so that would be picked up in the assay you were talking about, right? Because I've seen that with swabs where they didn't pick up enough, or they fell apart. And can you imagine that? It's
I know, and I agree, Johnny. And for me, it's still even though from a notified body perspective, it's not considered high risk. If the swab doesn't work, your assay is not going to work. So if you're not collecting enough DNA from the patient to be able to amplify that DNA and then test the virus that patient has, and there's an issue with the swab, that issue needs to be investigated for sure. But from a regulatory perspective and the approval at the notified body, they usually pick that up via the assay, which is the thing that links the patient and the result. And then the notified body will ask questions on the validity of that swab, but by virtue of the assay of the test that swab is used with. And what I would say in terms of clinical performance, so irrespective of, in certain cases, clinical performance not being applicable, a justification is always required. So the notified body just will not take, for instance, I've seen a lot of reviews when I was with BSI that were submitted as clinical performance is not applicable to our device. And that statement is not sufficient. So we need to provide a justification as to why it's not applicable. So that's really, really important. Because even if it may seem obvious to the manufacturer, the notified body will still need to justify in their conclusions and in their report what is the rationale behind clinical performance not being applicable. But there are certain cases where it's appropriate to say that it's not applicable if that swab is not on its own leading to results that correlate to a particular clinical condition, and whether that can be measured by an actual assay, for instance, that provides those results.
Well, Carlos, in our defense, you did say if we had any questions, Marie,
Yeah.
Hi, Marie.
and she's
Hi, can you hear me okay?
Yes.
Yes.
Hi, so Marie McClary, I work for CDMO CRO with a focus on the US market. A colleague in Europe let me know that there may be a pathway opening up for US medical devices to enter through Switzerland due to the shortage of notified bodies in Europe. Can you speak to that?
Yeah, of course. Marie, so I hear a bit of background. Oh, it's okay now. Marie, so one of the things that's happened is that Switzerland have decided that they are going to be accepting the FDA. So the FDA could be an entry route to Switzerland, but that does not mean that once you enter Switzerland, you can then access the rest of the European market. So you would be able to access Switzerland because of the a lot of issues. There was what they call a mutual recognition agreement between Switzerland and the rest of Europe, and that didn't go in time and so on. And with all of the bottlenecks with notified body capacity, Switzerland has decided that for their population, so they didn't want the Swiss population to be affected by the lack of notified bodies and potentially drop out in the number of medical devices and in vitro diagnostics available on the market. So they've decided that for the Swiss population, they would explore the FDA as an appropriate entry route. But that would just give you access to Switzerland, not to the rest of Europe.
Have they finalized that yet, Carlos, or is that still just a proposal?
I think it's just a proposal at this stage, Michelle. I haven't seen the latest, to be honest. I've been focusing on so many things that Switzerland has been off my radar. But there was a proposal, and I think that's really going to move on. I'm not sure if it's been finalized, to be honest.
There are some other questions, but I'll encourage you to go to the next slide.
Okay. So, and then what I wanted to talk next is just clinical performance in certain special cases. So it's very similar to what I was saying, where, so for example, a lot of cases in the IVD world, you've got IVD instruments, which is the machine that runs your test, controls, calibrators, DNA extraction kits, and that sort of thing. And in a lot of these cases, I've seen manufacturers demonstrating clinical performance as a combined approach. So they typically focus on the assay. So they don't say that clinical performance for my instrument is not applicable. They say it is applicable, but it's demonstrated via this assay, this test, and here's the documentation to support it. So in a lot of cases, clinical performance can actually be demonstrated as a combined approach based on certain devices. And the example are controls, calibrators, IVD instruments, incubators, and those sorts of IVDs. Now, if we look at, and this slide is a really important slide because it essentially establishes what are the sources allowed under the regulation to demonstrate clinical performance. And there are three main sources that IVD companies can use to bring products to market and to demonstrate clinical performance. And the three main sources are clinical performance studies. So that's when you actually do a study in a hospital environment or in the intended use environment, and you generate clinical data, clinical performance data for your device. Scientific peer-reviewed literature is one of the other sources. So this is very common, for instance, with devices that were legacy under the previous IVD directive, and now under the regulation, the manufacturers are demonstrating clinical performance based on articles and peer-reviewed literature that has been published around their devices. And then there's a third potential source, which is something called data from published experience gained from routine diagnostic testing. So that's the one that there's still a lot of debate around what does that mean? What does published experience gained by routine diagnostic testing? But for those of you on the call today familiar with the IVD world, it's understood as being data from, for example, external quality assessment schemes or proficiency testing. So anything that documents the routine use of that test and is published, and published means having accessible to the public with an identifiable source, could potentially be used to demonstrate clinical performance. So one of these three, as a minimum, should be one of the sources used. And the big challenge a lot of the times for devices that have been on the market for a very, very long time is that manufacturers cannot find any data that fits in one of these categories. And that makes the notified body review really, really difficult because at least one of the sources used to demonstrate clinical performance could come from one of these three. And then there's another option, and the other option is something that's called other sources of clinical performance data, but that cannot be used as a primary source. So that can only be used to complement. So if you've got gaps, for example, in your clinical performance studies, you can use things like as post-market surveillance data or old studies to support it, but on its own, other sources of clinical performance data cannot be used in isolation to support clinical performance. So the first three are the most important ones. And what I typically see is a combination of methods. So manufacturers will have, for instance, scientific peer-reviewed literature, and they'll have other sources of data, such as PMS data or a poster presentation that was done with documenting the routine use of the test or something like that. So those are some of the things to take into account. And it's really important because when you're defining your performance evaluation plan, which is the starting phase of clinical performance, you need to define up front, this is how I'm aiming to demonstrate clinical performance for the device. And you need to word it in a way that fits in one of these three main categories for it to be accepted by the notified body.
So now this part does sound like MDR, because this is exactly the same trouble that everybody's going through. Like, what are you gonna, like, what kind of clinical performance study are you gonna get, or scientific published literature that talks about a scalpel, or some basic
Absolutely
nasal cannula,
Yeah. And it's true, Michelle, we've got for IVDs, we've got devices that have been on the market for 20 years and they've never changed. And the studies were like, all manufacturers cannot even find them. The company was bought 20 times and by then, and the devices on the market, it works, everyone is buying them, but there is no data to support it. And that's where there's a real challenge with some of these older devices. Now with under the IVDR, there's no grandfathering, so any reviews that are submitted, the data needs to stand up to scrutiny. So PMS data could help, but one of these three sources should be available as a minimum.
I know that's probably a little early for this, but I know under the MDR products, they're already experiencing shortages of particular products, especially for pediatric applications. Is the IVDR having a similar impact yet? Because I think it could result in even more shortages than the MDR.
It could. I think so. Right now, no, because we've got this transition phase for the different classes of IVDs over the next 5 years. I think 5 years or 4 years, I can't remember, but it's up to 2028. So the first cutoff deadline is going to come next year, I think in May 2024, when the Class D devices need to transition. By then, if they are not all approved, that's when we may start seeing some shortages of devices. But at the moment, it's still as it was before, because we've got these sort of new deadlines that allow for a progressive rollout. But to be honest, under the new IVDR, it's really unknown the number of devices that need to move on to the new regulation, because the vast majority of them were self-declared by manufacturers. So we don't know exactly how many need to transition, but we know there's a lot of them. But in terms of numbers, I think that's largely unknown at this point. And I think the real issues are going to start coming apparent next year.
Carlos, Sanjeev is next, and he privately wrote me, hey, can you get me in touch with Carlos after this thing? So you may need to hire more people. What is what's the best way for folks to reach you after this presentation?
I'm very active on LinkedIn. So I'm Carlos Galamba on LinkedIn. So if you can reach me on LinkedIn, or I can put my email in the chat if that helps. And
please do.
Feel free to reach out.
Meanwhile, Sanjeev, what is your question?
Yeah, so Carlos, do readers come under the IVD protocol, or do they need to go through the clinical trials, or is it just the assays?
Sorry, say that again, Sanjeev. I missed the first part.
The readers which are there for IVD,
readers,
right? Do they need to go through the clinical trials themselves, or is it a combination?
The readers, do you mean the instruments or
correct.
the bit?
Correct.
Okay, so they need to be part of the clinical trial. So when you're doing a clinical study, or the right term is clinical performance study under the IVDR, you'll typically be testing your assay. But your instruments and readers are part of that study. So you need to define that this assay was tested with instrument X, Y, and Z. This is the version number of the instrument that was tested. The difference is that the instruments most instruments, 99% of instruments, they do not go through notified body review because they are Class A devices. Only a small portion of instruments will be reviewed by the notified body if they do not have an assay related to. So if they have something that's called an independent measuring function. So that's when you have a reader, for instance. This happens a lot of like in when you're measuring full blood counts, for example. Sometimes you have optics from an instrument that go through a sample and then provides a reading on that sample without needing any extra assays to be run. So in those cases, you would need that device would take the classification of the intended purpose, and it would be reviewed by a notified body. But yes, they are all part of the performance study for the IVD as well.
And would the data from, let's say we do a clinical study in the US, would that data apply, or would we have to do a clinical study in Europe itself?
So it's on a case-by-case basis. So at the moment, there's nothing in the regulation to stop you from using US data to support applications to EU notified bodies. But it's really important that you demonstrate that the US population can be seen as equivalent to the EU population. So it's on a case-by-case basis, depending on your test and the condition that you're detecting. What is essential is that you demonstrate and you have a justification as to why your US population can be considered equivalent to the European population. So in most cases, it is okay, and I say this with caution, but one area where I see that being a real issue is, for instance, when you have near patient tests, because in a lot of times, what is considered the near patient test user in Europe is different from a near patient test user in the US, because they have different qualifications. They have the actual people that can handle near patient tests, for instance, and tests done at the point of care can be different between the two jurisdictions. So you would need to make that argument if you wanted to use US data. In all cases, you need to make the argument, but just think of two things. Is the condition that you're testing in the US and the populations, can they be considered equivalent? And what are the conditions of use? So the near patient tester in the US equivalent to a near patient tester in Europe? And if you've got, you may have good reasons to say yes, but it needs to be demonstrated by the manufacturer.
So one follow-up question on that. Would in relation to this, would a multi-centric, multi-country trial be helpful in that case?
So a multi-center would be helpful when for a lot of areas. So for example, in the area of rare diseases, sometimes you need multi-center trials to be able to actually get enough data for your devices. So in a lot of the cancer type areas, you've got very rare diseases where you may find 6 patients in the US, another 5 in Europe, another 3 in Australia. So multi-center trials, they are good to provide more robust data for your submissions. So that could be one area. The other thing to think about is that for the US, and I'm not a US expert, we've got people in the team that does that for the FDA, but you always need a percentage of your testing done in the US. So for instance, if you're a European-based company, but you want to launch your FDA 510K, you need a proportion of the samples done in the US. So in that case, you may have the need to engage both US sites for your clinical trial and EU sites for your IVDR submission. But there's a number of reasons. There's cost reasons as well. You know, trials in the US are a lot more expensive than they are in Europe. So a number of factors to take into account.
Great, thank you so much.
I have a quick question, Sanjeev. You mentioned reader. Are you referring to lab equipments as a reader?
Correct. I mean, these are. I mean, in our case, we are developing the reader, the instrument, as well as the assay, right?
I see. So, question for Carlos then. I have a client where they have justified their lab equipment as non-medical device, non-IVDR.
Okay.
Because it either doesn't touch the samples. So for example, pathology samples, this machine will dry the samples so it's easy to cut. And they have justified it that neither it is a device nor IVDR. Regulation applies. But so it is interesting to hear this conversation. Maybe I'll follow up with you if it is going to take too long, but I would love to hear the answer.
Yeah, no, that's a good question. Well, I think it all depends on, so for you to classify, and I'm not entirely sure about the device, but if it depends on whether it fits the definition of an in vitro diagnostics according to the regulation. So there's a clear definition that says, controls, calibrators, equipment, apparatus, and stuff like that. So if they were able to look at that definition and it does not fit the definition, maybe there's a way. But I find it strange that something used to prepare samples would not be considered an IVD, or at least an accessory to an IVD.
Okay, I'll check that out. Thank you. So you're saying that there is equipment justification there. I'm not an expert in IVDR, so I have to check it myself. Okay.
That's fine. There may be a reason, but it's all comes down to the definition of what an IVD is, and we would need to look at it. Look at the device. What does it actually do in the lab, and is it, because if it's something that's used that at the end is going to help an IVD achieve its intended purpose, it could be an accessory to an IVD. So, and that falls under the IVD regulation. So,
okay,
that's good. I think it's we would need to look at it to provide you more information.
Okay, thank you.
Yeah, no problem.
Carlos, I just added your LinkedIn, your email, and your phone for the group.
Perfect. And I'll put my company email as well in the chat.
It reveals here that your given name is Andre.
I'm Carlos Andre, but my email is Andre, but everyone calls me Carlos. So, and I'll put the other entities here, the other email, the company one. Perfect.
Yeah. Great.
Okay, so what okay. Oh, so the only other thing I would say, and this is a question that we see happening quite often. So it's clinical studies. A lot of the clinical performance studies were done pre-IVDR, so before the IVDR was in place. In a lot of cases, they will not meet the definition of a clinical performance study under Annex 13, Part A, because that has a list of several requirements that need to be met for a clinical performance study to be considered a clinical performance study protocol. So it's a bit ambiguous whether, in a lot of cases, whether you can use that data as a clinical performance study. So you would essentially need to do a gap analysis on your study and see if most requirements of the IVDR are covered. If there are significant gaps, you may not be able to use that clinical study for an IVDR submission under this source of data from clinical performance studies. So just something to take into account. And now let's just look a little bit of some of the parameters for clinical performance. So the indicators of clinical performance, they may vary, and they depend strongly on the intended purpose and the performance claims of the device. So this is where you're going to be looking at things such as diagnostic sensitivity, diagnostic specificity, hazard ratios, and so on. So for example, if you've got a test that's claiming a diagnostic intended purpose, you're probably going to be looking at things such as diagnostic sensitivity and specificity. If you have a test that's claiming a prognostic intended purpose, you could be looking at things such as hazard ratios. So for instance, a troponin test, which is typically used for the diagnosis of acute myocardial infarction, it may require measures such as diagnostic sensitivity, negative predictive values, positive predictive values. For a test like CRP, which is a general marker that can be increased or elevated in a number of conditions, from inflammation to autoimmune diseases, then you could be establishing your clinical performance with method comparison. So agreement with another state-of-the-art method for CRP measurement, that could be sufficient. But depending on your intended purpose, that same CRP marker, if you're claiming that it can be used as a prognostic marker to determine the clinical course of a septic patient, for example, which is another indication for CRP, then your clinical performance will need to demonstrate additional parameters. So it will need to be linked to that prognostic value of CRP, of measuring the clinical course of a patient with sepsis. And your parameters would be slightly different. So you would be looking at positive likelihood ratios or negative likelihood ratios, for example. So what I would say is that there's one GSPR 9.1b, that provides a list of a number of clinical performance parameters that could be applicable to IVDs. And as a minimum, those defined in that list should be covered by the manufacturer. And if they are not applicable, there should be a justification. But remember that the IVDR is not exhaustive. Those, as a minimum, need to be justified. So for instance, if diagnostic sensitivity is not applicable for your device, you need to provide a justification. but there may be a lot more parameters that are applicable to your device and are not covered in that list under the IVDR, and that's totally fine. So the notified body will still accept parameters that are not called out in that particular list. So what are some of the gaps that the notified bodies are seeing? And we see this through consultancy as well. So we've got Article 56 of the regulation that says that manufacturers need to justify the level of clinical evidence that's necessary to support the safety and performance of a device, and that shall be appropriate in view of the device and its intended purpose. So one of the key things that we tend to find when we look at technical files for the first time is that the performance evaluation plan does not cover all of the elements in Annex 13, Section 1.1, and it's often missing a state-of-the-art description, parameters for assessing the benefit risk ratio, the development phases of the device, and how they link to the scientific validity, analytical performance, and clinical performance. And that's really the way you need to approach it is the IVDR is actually very prescriptive. So as you're putting documentation together and technical files together, just go through it line by line. So because if there's a requirement there that's missing, you'll get questions from the notified body. So you'll need to address it on a very thorough basis. Annex 13 has a list of things that need to be part of a performance evaluation plan. And anything that's missing, you'll get questions back from your notified body. Also, we've seen a lot of times you've got a performance evaluation plan, and then the output of that is a performance evaluation report. But the two, you can clearly see that they were prepared by two different teams, and they tell a completely different story. So what was planned in the performance evaluation plan looked very different from the conclusions that were done in the performance evaluation report. And that's typically the case when you've got broad or intended purposes that are unclear, because they are very difficult to demonstrate when they are unclear. Understanding of post-market performance follow-up is also an area that often leads to some confusion. So, for example, in your performance evaluation plan, you'll need to be considering PMPF already and defining what are the general and specific objectives of the PMPF activities. And in a lot of cases, that needs some rework before it can be submitted to the notified body. But most gaps really and I was in a recent BSI webinar where this was reinstated as well, is that most gaps that the notified bodies are seeing are in clinical performance. So they account for over 40% of the gaps, of the questions that they ask of manufacturers. And it varies from how they're applying the different sources of clinical performance data, what legacy studies that are being presented as IVDR clinical performance studies when they clearly do not meet the requirements of an IVDR clinical performance study, data that does not support the use setting. So think of if you've got a device that is intended for near patient tests, so it's intended for point of care testing, and all of your data was generated in the lab, you'll be asked the question of your notified body. So you're not generating data to support the intended purpose of your device. But we've got other things such as clinical performance data in the IFU that's not traceable, insufficient data for legacy devices, and those are some of the most common areas that we tend to see. Okay. So how are you doing for time? Because I don't see a time clock anywhere, but are we still okay?
We're okay. We go until we stop, and
Okay.
It's recorded. People drop off as they need.
Perfect.
But I recall seeing you have 25 slides. I don't know what slide we're on.
I think we are on, I don't know, but we should be near towards the end. So there's probably 3 or 4 more slides till
Okay.
we're done.
And the
But what
important thing, and I say this, all the time, you've clearly in this time demonstrated your vast knowledge and expertise. These next 3 or 4 slides may be incrementally informative, but what everyone's going to walk away with is, if I need this, I know a guy. And that's really the purpose of these. We get to know one another, what our strengths are, and how we as a team can be better than we would be on our own.
Absolutely.
So share as you like and conclude as you like, but I think we met a new friend today, and I appreciate that.
Thank you. Appreciate your kind words, Joe. Okay, so some of the lessons learned on clinical performance. So there are two ways I look at it. So we've got the legacy devices, so devices that have been on the market for potentially a very long time. They predate IVDR, and we've got our novel devices on the other side. So For legacy devices, really, when you're putting documentation together, make sure that there's clinical data that supports the full range of the intended purpose. And when we talk about the full range, is all of the specimen claims that you're claiming. So for example, for SARS-CoV-2, you may have nasal swabs, you may have oropharyngeal swabs. All of those specimens will need to be demonstrated, all of the clinical conditions claimed, and all of the target populations. If you have never performed clinical performance studies for your device, make sure there is a very strong justification for not performing IVDR clinical performance studies, and at least remember that one of the other two sources should be available, such as scientific peer-reviewed literature or published experience from routine diagnostic testing. Do some sort of gap analysis of studies that are legacy. So if you've done studies that predate IVDR, look at them again against the requirements of IVDR Annex 13 and provide justifications for any requirements that are missing. The other thing to take into account is equivalence under the regulation is something that exists, but it's very rare that notified bodies accept equivalence as a route for bringing a device, an IVD, to market. Because typically for IVDs, you're looking at chemistry, you're looking at instruments used with that IVD, you're looking at DNA profiles, and it's very difficult to justify how one device is exactly the same composition of another device. So as soon as you change the chemistry of your IVD, you're talking about a different device, and you need to have data to support that new version of the device. On the other hand, if your device is novel, obviously think about that notified bodies will expect clinical performance studies. So a new device to market could have a clinical performance study planned for it. And if you don't, you would need a very strong justification as to why that's not required. And also make sure that there's a clear link between scientific validity and state of the art for your novel devices. So we'll need to show that your marker is associated to a clinical condition and what is the state of the art in Europe for detecting that clinical condition. So most notified body refusals, if you get a refusal, it's typically because you've got significant gaps in state of the art and clinical performance. And one other item that's really key, and I see a lot of manufacturers missing, is if you've got a device, if you're lucky or unlucky, depending on how you see it, if you got a Class D device, which is like the high-risk IVDs, like an HIV test or a test to screen blood donations that could have a public health impact. There's one thing that is a super valuable tool at the moment, which are the expert panel views that have been published by the Commission. They are publicly available. There are 15 or 16 views that have been published so far on these very high-risk devices. The reason why that's important is that the views expressed by the expert panels essentially set what is considered state-of-the-art for these devices. So if you've got devices that are similar to the devices that went expert panel review and that are publicly available, you should be reading those reports to make sure all of your clinical data, all of your analytical data covers all of the gaps that the expert panels have seen in similar devices. Because if they've identified a gap in a device that's similar to yours, when you get scrutinized by the notified body will make sure that that gap that the other device had is not going to happen again in your device. And it's really eye-opening. So for instance, from one of the views published by the expert panel had to do with Epstein-Barr virus, where the expert panel challenged studies that did not include samples collected from organ donors and recipients, and it was very difficult for the expert panel to follow up and determine the immune status of those organ donors and to prove the intended purpose of the device. For SARS-CoV-2 assays, they've queried that in one of the cases there was insufficient evidence with anterior nasal swabs. There was another one with Ebola where the test was done with spiked samples, so not real Ebola specimens, and the manufacturer did not conduct any studies during the latest Ebola crisis in 2014. So those are the things you need to think about. If there was a question raised by the expert panel on a device that's very similar to yours, make sure you've addressed it in your technical documentation. Otherwise, that question will come back from your notified body once your next review goes through. I think there's a question from Zoe.
Hi, yeah, you just mentioned spiked samples there, and I was wondering if for things where you have a really rare target, or maybe a novel target that isn't necessarily routinely tested for, so it's really difficult to find existing samples. Have you experienced any evidence of notified bodies accepting contrived or spiked samples in those situations in a clinical study?
And Zoe, do come on camera if it's at all possible, so we can see you.
Oh, okay.
Yeah.
Hi.
Zoe, it's one of the lovely answers, right? It depends. So personally, in my experience, I've seen that happening. I've seen it being accepted, but you need to have a very strong justification. And the rarity of the disease could be a strong justification. But what you'll need to think about is how you're going to close that gap once the device is on the market. So you'll typically need to be committing to a strong post-market performance follow-up plan. If you've used partial of your study with spiked samples, you'll need to commit to a PMPF, a study, once the device is on the market, to make sure to allow you to give you enough time to gather more data on those more rare markers when they're not available. So for instance, one of the issues we've got at the moment, and we're doing a lot of studies with HIV devices as well, like finding HIV2 samples in Europe at the moment is almost impossible. And the common specifications require you to get HIV2 samples as you're trying to demonstrate your clinical performance. And it's very difficult to find them. So sometimes you'll need to think a little bit outside of the box. There could be, I've seen justifications working for using spiked samples, but typically that is accompanied by a very strong post-market performance follow-up once the device is on the market, with a clear study that this is how we're going to monitor that these spiked samples do not impact performance of our assay. We aim over the next 2 years to test these many samples and provide that evidence to the notified body. But I think it's possible to build a convincing argument.
Okay, thank you.
No worries.
Almost
Okay,
one more question.
Yep.
Can you do retrospective studies rather than having to collect new samples?
Yes, you can. So a lot of the IVD studies are actually done with retrospective samples. But so one of the things to take into account is that if you're using retrospective samples, it's going to have more bias in your study. So prospective sample collection could be the default option. But if you can demonstrate that using retrospective samples will not impact performance of your assay because the markers are quite stable over time, they do not deteriorate, after being stored, frozen, whatever, then you can use. They usually call it leftover specimens or archived specimens. So, and that would be a retrospective study design.
But I guess for readers, you can have retrospective study, right?
You can. I think it really depends on what. Why do we? A lot of cases, what we see is that sometimes you need samples that are fresh to be able to test the disease, and for that you need prospective sample collection. But in other markers, you have things like hemoglobin or whatever that, or full blood counts that you can archive a specimen, or even some tumors. They stay. You've got the FFP samples, in paraffin embedded tumor samples, and that's used all the time to study some of the cancer markers, and they are done with retrospective specimens.
Yeah, I guess for depends on the risk, is like how accurate or how closely you are with your your device or IVDR. But for software, and I think it applies to medical devices and IVDR, I have done retrospective study to show that software algorithms perform, AI performs to the standard, whatever that specificity, sensitivity you define. But yeah, this is excellent information. Thank you.
No problem. So the other area that I just wanted to focus on, is there any other questions? No. So the other area is just near patient tests and clinical performance for near patient tests. So near patient tests, so under the regulation, so everyone knows near patient tests as point of care testing, but then the IVDR decided to call them near patient tests. But it's the same thing. It's point of care near patient testing. But when you're defining it for regulatory purposes and in an IVDR file, use the right term, the right regulatory term, which is near patient testing. So really things that are important on near patient tests is that the instructions supplied by the manufacturers should make it clear what the level of training, qualifications, and experience of the user. So what are they? Are they nurses? Are they healthcare professionals? What training do they need to go through before they operate the test? It should be validated in appropriate environments. So is it a blood collection center? Is it a GP clinic or a sexual health clinic, an intensive care unit? So what you're gonna, how you define your intended use environment is going to be very important, because, for example, if your device is meant to be used in emergency care next to ICU or in accident and emergency, the study is going to look very different from a device that's done where the doctor is in a clinic, for example, because the conditions of the testing are going to be different. So in an ICU unit, you're typically going to be a lot more stressed. There's factors, environmental factors to take into account, that when you're in the comfort of a physician's office, those factors will not come into play. So just things to take into account. And also, we've got devices that have dual intended purposes. They can be used for laboratory or near patient testing, and the study should be designed to cover both. And obviously, particularly for near patient tests, usability becomes very, very important.
Carlos, I'll say that usually I say, we go as long as we can because nobody ever goes this long. There are never this many questions. And Stina has written a very comprehensive question for you to follow, but I need to retract that. I have to take my mom to the airport. I've enjoyed her visit this week. So now I'm getting a little stressed because you don't want to leave my mom waiting.
Absolutely not. Absolutely not. Joe, and what I'll do, I'll just go to some take-home messages, and this is my last slide. So very just to summarize. So when you've got clinical performance, make sure you define a very clear intended purpose. Look at your sources of clinical performance data and make sure they are well described up front in the performance evaluation plan. Studies for new to market devices or novel devices, so clinical performance studies, are typically going to be expected by notified bodies. If you're thinking of using equivalency, think twice. That's probably not the right strategy for your device. Think of what is the state of the art in Europe. Avoid use of terminology from other jurisdictions. So we've seen a lot of files with, they reference the FDA 510K and RX only, and terms that are very US centric. So a notified body will expect a technical file that is written for the EU. So just submitting the 510K file to a notified body won't meet IVDR requirements. And I think I'll leave the presentation there.
Well, I'll leave the presentation with, this was very well received, very much information, a lot of comments that we didn't even get to. And my gratitude on behalf of the group. You're welcome to come back any week. We meet every week, a different topic each week. And next week, a fan favorite, Rob Packard, is going to be our presenter. He's going to be talking about how to handle risk in your submissions and in managing risk. So if you can join us, you're more than welcome to do that. So on behalf of Joe Hage and Joe Hage Enterprises, thank you, everybody. I'll see you next week.
Thank you all. It was a pleasure. Thank you.
Bye.
Bye.